If there's no more announcements, I would like to thank all of you for joining us today again in the Microbiome Center seminar. And I'm pleased to introduce today's speaker, Dr. Martin Polz, a professor of microbiology at MIT. Dr. Polz is currently on a sabbatical at University of Vienna, where he also earned his doctorate of natural sciences in zoology before earning a PhD in organismic and evolutionary biology from Harvard University. His lab's research is focused on molecular microbial ecology and environmental genomics, with emphasis on population structure and differentiation. And today he will talk about how interactions structure microbial diversity from genes to ecosystems. And before we start, I would also like to mention that his postdoc, Dave Van Insbergen, will be giving a Microbiome Center workshop on analysis of gene transfer in microbial populations. And that's going to happen on October 8th and 9th here at Penn State. And with that, I will hand it over to Martin.
Yeah, thank you so much. I really wish I could be there, but this is great. And I don't know how you handle these kinds of talks, but if you feel like it's better to interrupt me, if you have any questions, feel free. I should say that I've actually moved to the University of Vienna, so I'm no longer at MIT. If any late stage grad students are listening, I'm looking for postdocs at the moment. And should there be any talented undergraduates listening, I'm also looking for PhD students. So if you want to work here at the University of Vienna, there's a really great microbial ecology center called DOME. So you can look it up if you wish. Okay, so my talk today is going to be about interactions. At least that's going to be the theme that's going to tie things together. I'm going to try and cover some relatively diverse topics, as you will see. But fundamentally, I think we're very well aware. I can't advance the slides for some reason. There we go. That'll work. All of us are very well aware, I think, in this audience that this is really a microbial planet. All ecosystems really are packed with microbes that fulfill really various important functions. There would really be no life on the planet, of course, without microbes, them having been the first ones on the planet and having shaped all the biogeochemical cycles. And we've gotten relatively good at counting them and figuring out who's there. But to really figure out who's interacting with who, I think is one of the big topics in microbial ecology at the moment. And so I'm just highlighting here these numbers just to remind everybody that the marine environment is, globally speaking, with about 500,000 to 1 million microbial cells per mil, at least in the surface ocean. And its large volume is one of the key microbial ecosystems, and I'm going to talk a bunch about that. And then also sediments and soils are tremendously important. And I always tell my students that's the big frontier. You should go into that, because I think the ocean is actually fairly well studied by now, but soil interactions, I think, is unbelievably challenging, and I salute everybody who's trying to figure this out. And then, of course, animal guts are an important ecosystem for us. Globally speaking, they're not so important. I always say in my lectures that animals fulfill the important role of chewing food for microbes, and I think that's kind of the way we can think about them, because they are fed by us, obviously, and but in return fulfill some very important functions. So when we think about interactions, we think about naturally about species or populations that interact with each other and that influence each other, be it through more or less direct interactions such as competition or predation, for example. But how these interactions are really structured is very poorly understood. And one of the things that I always tend to think about is it's become very popular to think about protein-protein interactions or gene-gene interactions. But think about a microbial ecosystem where you try to figure out interactions, but you wouldn't know what a gene is or a protein, because we have a very poor idea how to circumscribe really cohesively behaving populations in the environment. And so part of the talk today is going to be about that. So what I'm going to try and cover is some recent work, much of it not really published yet, on ecological interactions. And the first part is going to be on virus-host interactions in the wild and how these are structured, because I think there's a great deal of unknowns there as well. And then the second part is about whether we can actually define populations as units of ecological interactions. Okay, so let's jump into the first one, the question about the structure of bacteria-phage interactions. And I have to say this is going to be a series of tales to me that highlight how little we actually know about these types of interactions in the environment. And a key person in my lab who initiated this work was Catherine Kaufman, who was a PhD student and subsequently a postdoc in my lab. And so she took on this big question of how to figure out how microbes, how bacteria and their one of their main predators or putative main predators in the ocean, bacteriophages, are actually interacting. So I mentioned already that microbes in seawater are about 1 million or so per milliliter. When you try to count viral-like particles in the ocean, you come up with about 10 times more. So there's presumably about 10 times as many viruses as there's microbial cells in the ocean floating around. They presumably alter ocean carbon and nutrient cycling by lysing bacteria and also algal cells, as well as other organisms, and contribute thereby to dissolved organic carbon, nitrogen and phosphorus pools that then fuel microbial or primary production again. In general, it's thought that viral predation is highly specific, but bacteria are highly diverse. And I will say right away, after working on this topic for about 10 years now, I do not understand how a specific virus ever finds its host in the ocean. That said, I think we still figured out some quite interesting things. So how does all this killing actually happen? Counting doesn't really tell us. Sequencing does not tell us yet, because we can't really link viruses to their hosts. We can maybe link viral groups to presumed bacterial hosts. And so Catherine reasoned that we really need live bacteria and viruses to really figure out these interactions. And so she set out during her PhD to do something completely insane. She went out for every single day for 93 days in a row and sampled the coastal ocean. So for 3 months, every single day in the morning, she went out and finished by midnight processing the samples. I always said to her, take a break. And she said, no, if I do, I will never go back. And so we have this really fantastic data set now that consists of 93 days of 16S ribosomal RNA sequences, 18S sequences. She archived virus concentrates, microbial cells for each day. And on 3 days, she isolated about 1,000 vibrios, which is our model system, long-standing model system in the lab, and used 400 of the strains that she isolated to pull phages from the water column. And she did this by direct plating so that we have a fairly quantitative estimate of the viral abundance of the different viruses, I should say. And then she did another insane thing. She did a fully resolved host range matrix of the viruses that she pulled out. And it's about a 250 by 250 phage host interaction matrix that is now fully genome sequenced, where we sequenced about 1,000 bacterial hosts and over 300 phage. And I think it's to date, it's actually the largest virus host interaction matrix there is, at least that is genome resolved. So what she found was the following, that most viral interactions are actually highly specific. What you see here is a phylogenetic tree on the outside of the Vibrio hosts. You note that many of the strains are very closely related. Overall, the bacteria are very closely related, and that was actually key to the success of this, I think, because we can really see where the boundaries of specificity are. And so the dots here in this matrix are an individual virus, and the lines then connect them to their host. And you see that most of those dots are clustered on the outside, but there's a notable exception here, and a couple of other ones, but this big cluster here seems to have much broader host range. Note though, even though these have broad host ranges, they are still fairly specific for individual strains within the bacterial populations that we pulled out. And so I should also say that the color coding here, which will return later on, denotes population structure that we figured out for the bacterial hosts. So I want to first talk a little bit about this unusual virus here, which is a new, very generalist virus. It is unlike these textbook-like bacteriophages that you see everywhere that look like a lunar lander. They have the capsid with the tail. These are tailless phages. They are very poorly understood. We know that they exist. There's only a single isolate from the marine environment from the 1960s. There's a few more known for gram-positives. They have a very different capsid fold than the cauda-viralis, so those tail phages, and they're actually much more closely related in the capsid fold to many eukaryotic viruses. And the important point is that this virus actually resembles the morphological majority in the ocean. So people have actually said, when you look at viruses under the microscope, under the electron microscope, that 80–90% of the viruses that float around in the ocean are tailless. And so this, we think now, is one of the first examples of such tailless viruses in the ocean. They have some very interesting properties. They're very small. They're only about 10 kb in genome size. Most cauda-viralis start at about 50 kb, go up to a couple hundred kb oftentimes. They were only 7% of the isolates, but because of the broader host range, they were responsible for 37% of the infections. And I should also mention that there's a prophage version of this virus that is very, very broadly distributed amongst the phages, amongst bacterial genomes, I should say. Now, why was this virus not, or these types of viruses never found before? And it was really astounding because there were 4 factors that really conspired against these viruses. One of them is that they're killed by chloroform, because they actually have a membrane that surrounds the capsid. And if you add chloroform to phage preps, which is oftentimes done to kill contaminating bacteria, you actually kill these viruses. And that's shown in this graph here, where chloroform addition killed these types of viruses, but left cauda-viralis that are shown in white actually unaffected. One of the most amazing things actually to me was that these phages are also absent from metagenomes oftentimes, because they have a covalently linked protein attached to the DNA. And so if you do just a regular DNA extraction without proteinasing your DNA beforehand, you lose the DNA specifically to the protein fraction. That's shown in this graph here, where you see untreated and proteinase-treated viral prep, and you see that when you take it through regular DNA extraction, the untreated one is lost. Cimilarly, a lot of people will use density centrifugation to create concentrates of cauda-viralis, and you lose them because they have a lower density because of the membrane. And then also they are slow on plates, which is something that we haven't quite figured out why that is, but they come up only after about a week or 2 on plates. And so Catherine was so extremely careful, she kept rechecking her plates, and that's the only reason she actually noticed those type of phages. Well, most people probably throw the plates out after a couple of days because the cauda-viralis are generally very fast. When you actually now have a sequence, you can find them. You can find them, the prophage version, in different genomes. You can find them in various metagenomes that have been proteinased. And this is just gives you an impression of that. But they are hard to find because they're extremely sequence diverse. And so we had to actually employ a really labor-intensive iterative search where we added the sequences that we found to the search and did this over and over again, and finally ended up with a sizable sequence diversity of these phages. I should say though that we have found them in microbiomes now as well, so that might be of interest to you as well. Okay, so this is a new phage that came out of this screen. But the majority of phage are still very specific and only seem to infect very few strains. And so we were interested in this question, what actually determines this phage specificity in the wild? And so people oftentimes divide phage specificity and defense against phages into two types of classes: external defenses, which oftentimes means receptor modifications, which means that the host cell can actually receive a mutation that makes it renders it immune to phage attachment, and so the phage can no longer attach and inject its DNA. And these are invariably the type of mutations that you get when you throw a phage and a bacterial cell into culture together because of the strong selection. But there's also a whole slew of internal defense mechanisms. Famously, of course, CRISPR-Cas, restriction modification systems, then also perhaps lesser well-known abortive infection systems that oftentimes resemble toxin-antitoxin systems. But then in recent years, there was a whole slew of the new phage defense mechanisms that were discovered, mostly from a lab in Israel that has done a fantastic job to describe these. And so for us, the question was, what really determines specificity in the wild? Is it receptor modifications or defense genes? And it turns out that the dynamics in wild microbial populations are really very poorly understood. So going back to our matrix, which I now show in this form here, where we have on the side the host phylogeny using a protein coding gene, and here are different classes of phages that we pulled out. Here's this very broad host range phage that I just introduced, but here are all these cauda-viralis that I mentioned already previously. They look like very specific, and overall the matrix looks very sparse, which was actually a big surprise for us. Because we actually were able, because we used phage concentrates from the environment, we were able to have a detection limit of about 15 phage per liter, which is pretty low. So this may indicate that many of the strains are actually quite well defended against the phages that are currently present in the environment. And so one other feature that we saw, though, is that we oftentimes had these blocks of host strains that were killed by one particular phage, but then oftentimes very closely related hosts were killed by another strain, or by another phage, excuse me. And I want to zoom in into one example of that in a little bit more detail, where you see here this host block. This is fully genome sequenced. They only have 17 SNPs amongst each other. So by all intents and purposes, they're very highly clonal. Yet the phage that infect them are very different from each other. They're both cyphoviridae, so they have these long tails, but they're very unrelated. They can't really be aligned very easily with each other. So they're completely different phages. And so we were interested first in the question whether there's any receptor modifications that might actually determine the specificity. And we did a couple of different screens. We did transposon mutagenesis as well as resequencing of spontaneously resistant mutants. And in all cases, they mapped to the following loci. For this orange phage, it mapped to the general secretion pathway. And for the second phage, this purple phage, it mapped to the LPS, so the lipopolysaccharide, and then secondarily to sodium translocating NADH reductase. And so the idea here is that the putative receptor here in the first case for the orange phage, this general secretion pathway, that is most of the mutants map to this protein K, which actually sticks out and might be a phage receptor. And in the second case, we have two receptors, which is oftentimes the case for cauda viralis. They oftentimes attach to the carbohydrate coating on the cell, and then have a secondary receptor that is a transmembrane spanning protein, and through which they can actually inject the DNA. When we looked across all of our hosts, all of these genes were completely identical. So we concluded that the phage receptors are actually invariant amongst these strains and are not really determining the phage specificity in this case. What was really notable, though, was when we looked at the gene presence and absence amongst these strains, was that there was a considerable component of flexible genome, or genes that were not present in all of the strains. And when we hierarchically clustered these, we neatly got back the infection specificity. Okay? So this is the clustering of the flexible genome. And so we looked into this flexible genome component, and what we found was that both host groups are differentiated by the presence of putative mobile genetic elements that in all cases carry defense genes. And these are really, really interesting mobile genetic elements. They assemble very, very poorly in short read sequencing, even when you have quite well saturated sequencing runs. And so we had to do a long read sequencing to really assemble them and figure this out. And we think that's actually why they've been missed so far. But I think they have some very interesting potential biology. They insert usually in a very specific location in the host genome. In many cases, we found that they cut apart a particular host gene. But when they do that, they actually replicate the host gene inside the mobile genetic element. And then the other striking feature is that they have these defense gene. In this case, the genes, in this case, it's a restriction modification system. Here, it's a toxin-antitoxin system, another toxin-antitoxin system, and here we have two restriction modification systems. The rest of these things annotates extremely poorly. So we have actually very little idea what the other genes do. And this is actually important in another point that I'll make in a couple of minutes. So we think they can excise, and we think they can actually also package themselves into virion capsids if there's an infection. So we were interested in how they actually work, and we created some knockout mutants, knocking out systematically these three defense islands in the orange group of strains. And I should say that that was actually quite difficult because of the toxin-antitoxin system. So we always had to knock out the toxin first. And but we finally managed to actually knock out all different combinations of these defense elements. And so what I'm showing here then is the phenotype of these mutants. Here's the wild type, and here what you see here is a dilution series of the phage. And note the following, please, that at high phage concentrations, you oftentimes do get killing. This is a phenomenon called killing from without, and it's thought that this actually happens because when you have high phage concentrations, the phage saturates the receptors and pokes a lot of holes into the membrane, and therefore the cells still die. But in these cases, you never can propagate a viable phage, which is shown here on the right. Okay, this is a restreak of these lysates to see if we can actually get viable phage. Defense element 1, if you knock it out, you get a little bit of effect. You get more sensitivity to killing from without. Defense island 2 or 3 knocked out has almost no phenotype. 1 and 2 knocked out resembles knocking out 1. And then 1 and 3 knocked out, you get more killing from without. It becomes more sensitive. But only when you knock out 2 and 3 together can you then recover actually viable phage. And that's because that only leaves the restriction modification system in there, which is notoriously leaky. So every 100th or so infection is thought to actually can actually produce a viable phage because the methylation happens correctly. And when we knock out all 3 of them, we get basically the same sensitivity to the phage that the purple wild type has. Okay? And so here is shown the viable phage streaks. So what we think is happening is that the first level of defense is actually the restriction modification system. It provides a leaky protection. Viable phage can be produced at high concentration or every 100th infection or so it is thought. And PD2 and 3 are so-called abortive infection systems that actually kill the host. And so therefore you also get this phenotype where you get higher sensitivity, but you still never recover a viable phage. So one of them might just arrest the host, but the other one actually seems to kill the host potentially. Okay, so then the other interesting thing is when we looked a little bit more broadly, still amongst very closely related genomes, we found an enormous number and high diversity of these phage defense islands. They can make up to 90% of the flexible genome, and we think now that it actually explains a very, very large fraction of unannotated genes in the flexible genome. Because you know, whenever you look into these accumulation curves of the flexible genome, you see that it's ever increasing, and it correlates very well with these phage defense elements. And a single strain we found can have as many as 18 of these phage defense elements or islands. And we looked not only in our Vibrio system, but in Salmonella, as well as in Listeria, so it's a couple of pathogens. And we find ample evidence for these phage defense islands or elements as well. So what are the consequences about thinking about how viruses might actually control microbes in the wild? I think that this changes a little bit sort of how we might think about this very popular model that's called kill the winner. And the elements of that are basically that if you have a microbial community and some genotypes in that community receive a growth advantage for whatever reason, either because of evolution or ecology, they could proliferate and ultimately they might outcompete competitors. But it is thought that viruses can respond very quickly and suppress these successful genotypes, eliminating them, and thereby in the long run actually preserving microbial diversity. Now, what we think though, and we actually postulated this in a review, Otto Cordero and I, some years ago, but we were thinking of receptor genes actually being hypervariable and being transferred very much. What we see is essentially that phage defense is completely unlinked from any other property in the genome. And so it's really a game between these phages and these phage defense elements, at least in these relatively low predation systems. I don't know what happens if you have a clonal bloom, for example, of a pathogen that you might hit with phage and phage therapy. That might be a different situation, but in the natural environment, I think this is a game that has been played for a very long time, and it is largely between these phage defense elements and the viruses. And the hosts are really just a host to yet another mobile genetic element that takes advantage of them. I will say, though, that I do think this does have some implications for phage therapy, for at least prospective phage therapy, because it does show that phages of resistance to phage can actually be just as easily acquired as antibiotic resistance, because it sits on mobile genetic elements and travels amongst clones very freely. Okay, so with this, I'm going to switch topics a little bit and talk about, did you have a question?
Yeah, I do. I have a question about the very last statement. I think before we move on, it might be a good place for it. There might be other questions, but so, you know, one of the ways people have gotten around resistance development with phage therapy is that they have generated resistant cells to a particular phage, then isolated another phage, generated that so it's resistant to that, and then isolated another phage, and then created these phage cocktails that would kill one genotype, let's say.
Yeah, no, I think that's, I don't think phage therapy in sort of when you have a particular bacterium that you isolate from a patient, you can isolate many, many different phage that will hit that bacterium. But I'm talking about a sort of a more a forward-looking phage therapy where you are trying to assemble phage cocktails for the long run, which has been attempted in various settings. But if you have a patient who suffers, you know, from an antibiotic resistance infection, and you can isolate this, and it's highly clonal, I think you can easily isolate phage that will kill this bacterium. That's a very viable strategy, I think. But a more forward-looking, a therapeutic that has you know, broader application without exactly knowing the genotypes, and for longer-term treatment amongst the patient cohort, I think it's very risky. It bears the same risks, I believe, as antibiotic resistance.
Thank you. That's very helpful.
Yep, I have a question, if possible. Yep, it's very interesting, very interesting topic. Thank you very much. The question that I have: there was a mechanism, another mechanism that called suicide mechanism. Suicide bacteria, the infected bacteria, like victim itself. Did you look up to any of these studies or something? Or
I'm sorry, I couldn't hear you very well.
I'm saying that there is a mechanism that some researcher talked about. It's called suicide mechanism. The infected cell will kill itself. I mean, beside the modification system or the CRISPR mechanism, or there is a suicide cell in which the cell kills itself, and then the phage will be killed as well.
I think that's exactly what we see in this abortive infection.
Okay.
Yeah, this is what I meant by that. So abortive infection can have two manifestations. Either the cell manages to arrest the replication and deal with the phage somehow, or it can commit suicide.
Okay.
And I think we see both elements of that.
Is there a specific mechanism or detail about this mechanism, how it's work? This suicide mechanism?
Yeah, it's these toxin antitoxin systems. So abortive infection, ABI systems, they're called.
Another question, sorry. You have mentioned that one of the for some phages it took 2 weeks for them to replicate. I mean, 2 weeks to see the clearing zone on the plates, or 2 weeks?
Right.
To, Wow, it's very. I mean, this because usually most of people throw their plates after. So how about after a week maximum? 2 weeks is still long. So how would how's the license activity of these phages? I mean, after you got them in 2 weeks, how you would be able to propagate them or get them in a large number with this?
Yeah, I mean, you basically pick you know, with a sterile toothpick that you put into the clearing zone, into the plaque, and that is usually sufficient, and then re-streak it onto a plate. And after you do this a few times, you have a presumably pure phage, you know, which you can test in various ways.
Yeah, and every time you have to wait 2 weeks. I mean, even after.
So there's quite a bit of variation amongst these phage. Some are faster, but by and large, yes, they're slow.
Oh wow! Yeah.
Yeah. Yeah. We don't understand why, and we know very little about this phage. To be honest, we're working on some aspects. We don't know how it enters the cell, for example, and one of the hypotheses, which is kind of intriguing, because you wonder how such a phage could actually compete with these fast-tailed phages, right?
Yeah. Yeah.
And so one of the hypotheses that we have is that it might actually be a specialist for cells that are non-growing.
Well, interesting.
That it can actually, which tailed phages, once they infect a cell that doesn't grow anymore, they just sit there, right? So that's
you mean temperate phages or just any type?
No. They inject their DNA into a cell that's not growing. They will not replicate.
Okay.
But these phages might. We're not sure. I mean, this is a sort of out there hypothesis that we don't have much to support, except that they take long and seem to do well on plates where, you know, nothing seems to be growing anymore.
Okay.
Martin, if I could ask a follow-up question.
Sure.
This is Tom Wood. We discovered the relationship between toxin and antitoxins as phage inhibitors in '96.
Right, yeah.
And since then, we've recently written that there's very little evidence that toxin antitoxin systems are related to cell death of the host. So with your PDI 2 and 3, you think they're abortive inhibition systems, which I think have been related to cell death, or do they definitely include toxin antitoxins? And it would be interesting to me because you said it, you know, at least for one of them was actually killing the host.
Yeah, we think that's happening. Yeah. So, well, they came back annotated that way. We have not done much in terms of characterizing them more.
Okay, thanks.
Yeah. So
I don't know if I can ask another question.
And we find a ton of these things, you know, oftentimes in these mobile genetic elements, and sometimes it's only restriction modification systems.
No, it's beautiful because, you know, it's quite controversial what the TA systems do, but the only phenotype that seems to be hanging on at this point is phage inhibition.
Right. Yeah, and I think that's super interesting because that would also explain the great diversity, right?
Yeah, so thanks.
Well, I'm sorry, I don't know if I can ask another question or not now.
Sure.
Yeah, sure. Among those all the strains that you have, like 480 strains of bacteria and maybe 1,000 samples, did you find any strains that was resistant to all phages? I mean, you was not able to isolate any phages for them at all?
Yes, most are resistant to all phages.
Oh. Yeah, no. Well.
That's what I said is the sparsity of the matrix. It's a fully resolved matrix that I showed before.
Yeah.
And most are resistant to all of the phages.
So you were not able to isolate any phages to this specific strains.
Yeah. So remember we had some 3,000 bacterial isolates.
Yeah.
And we tested. I can't remember how many were really included in the matrix now, but a large number, and most of them are actually resistant to all phages. So that this is the white in the matrix.
And do you think that's because
I do not understand how a phage finds its host in the ocean. There's something we fundamentally do not understand.
It's not actually in the ocean only, it's everywhere, also in the microbiome.
Yes, absolutely. I think there's some spatial and temporal clustering that needs to happen in order to.
Okay, we may need to do a further discussion later. I might contact you.
Sure.
For the sake of time. Okay.
I'm not in a rush. Okay. So the second part of the talk is basically about something that my lab has been working on for, I think, a good 15 years. And this is trying to really define what populations are. And, you know, when you pick up a biology textbook, of course, it's also, the biological world is so neatly categorized into species, where species really are reproductive units, which means a gene flow unit. And then there's populations, which are local representatives of a species separated by gene flow discontinuities from other populations, perhaps. And then these gene flow boundaries ensure that adaptations can spread in a private manner to either a well-isolated population on a temporary basis or within a species. And so the problem is that for microbes, even though we use these terms all the time, we really lack an effective definition for species and populations. And the reason, of course, is horizontal gene transfer, which people have said is absolutely, you know, incompatible with even the notion of a species. But I disagree a little bit, as you might guess in the next few slides. So we have, of course, I referred to this already, we have oftentimes very large gene content differences. This is a very classic example of 3 E. coli strains that were sequenced, where you have only 40% of the genes are shared between these strains, and the rest of the genes are either shared by a couple of the strains or are even private to a single isolate. And we know of many, many such examples and studies now. This is a pervasive pattern in microbial diversity. And as I mentioned before, I think that these phage-defense elements might be at least a large portion of this unexplained diversity. But then there's also recombination, homologous recombination, that really scrambles phylogenies. And so this is an example here where you see that you use different genes in a group of microbes, and you get different clustering. And so phylogenies are notoriously unreliable for categorization of microbes. And there is a tendency to use whole genome phylogenies with the idea that if you average across the entire genome, you might get a more reliable majority tree. But there's some recent work that actually shows that that might not even be true, and I'll show an example of that in a little bit as well. So what we use then are completely arbitrary clusterings of microbes, categorization of microbes, oftentimes in ecology still based on the 16S ribosomal RNA gene, which has very poor resolution. Especially when you take tag sequences, you get maybe to the family or genus level. And in epidemiology, there was a tendency to use multi-local sequencing approaches, and now increasingly whole genome approaches. And it was actually that that I initially tried to transpose to microbial ecology, these kind of approaches that were pioneered in epidemiology. And I think it led to some progress, but still it was very difficult to actually find some consistent patterns. So the first thing that we tried to do was basically to incorporate not only genetic structure, but also habitat associations in the environment. And this was a collaboration with Eric Ahm at MIT quite a few years ago now, where we developed a model that we called AdaptML, where we postulated that a microbial group or a population would occupy a particular habitat in the environment. So it's associated with ecological factors, if you will. This is an unknowable variable, though, a hidden variable. But what you can do is you can differentially sample the environment. You can categorize samples, and you can hope that your habitat structure is differentially distributed amongst your samples. So that's something you can measure, and then you can construct phylogenies, and you can map which sample your individual strains were isolated from, and then you can use modeling basically to see when one characteristic habitat distribution changes from, or characteristic habitat distribution changes from one to another. And so that gives you not only an association, but also a sort of a history of association in a sense. We did this for our model system, the Vibrios and microbes in the water column. Any water sample is really a universe of ecological opportunities. You can be free living, you can attach different kinds of organic particles and hydrolyze them. You can associate with different algae or smaller animals, and then there's bigger animals, surfaces, and so on, and everything moves around. So there's a lot of transfer between these habitats. The way we approximated the water column structure was by using size fractions, and this was actually very successful. So the less than 1 micrometer fraction would be definitely free living. Then the 5 to 1 micrometer fraction would be mostly free living or attached to very small particles, and then the larger size fractions would be attached to different particles or organisms. We don't know. But when we did this and characterized about 1,000 Vibrio isolates initially, we saw that we got some very, very fine scale habitat distribution and differentiation. And we called these putative populations. And I should say that we followed up with different approaches, and in almost all cases, this actually turned out to be quite robust. So the lesson from that is that these are protein coding genes that are shown here. The 16S ribosomal RNA would resolve almost nothing of this structure. The biggest sort of surprise and also confirmation was that we were actually able to reconstruct a speciation event of one of these clades that was shown in this phylogenetic tree. So focus on this one here. You see that you have a group of very closely related strains, and they're distributed amongst the green and the red size fraction. So there's a yellow size fraction in between. So there's very unlikely that there's bleeding across. So we thought that these would be a good one to actually target with next-gen sequencing and trying to figure out what the genome dynamics are. And the reason I'm bringing this in a little bit more detail, even though it's also a little bit older now, is because it really put us on the trail of how to estimate gene flow amongst microbial populations. And so when we did a whole genome phylogeny, what we saw was that we recapitulated the habitat distribution quite well amongst the strains. But Jesse Shapiro, who spearheaded this work, was also curious about how recombination was affecting the diversity across the genomes. And so this is a bit of a complicated slide, but what you see here is on the x-axis, the 2 chromosomes that Vibrios have. So these are the chromosome positions. And on the y-axis, you see the SNPs, the number of SNPs, so single nucleotide polymorphisms that support this ecological split that we just saw in the whole genome phylogeny. And the thing to notice is that the support across the genome is basically 0. Okay? And this is shown down here in individual trees. These are recombination blocks, so that have a unique recombination history across the genome. And the most abundant one, this is a little bit cut off here, the most abundant tree that we obtained was 0.7% of all the trees. So it looks completely mixed, except for some regions here that are shown in these bars that contain the majority of the support for the ecological split. And it's obvious why that is the case, because in all of these cases, the green population had much reduced diversity. So it was, in almost all the cases, it was monomorphic in these regions. And it sits on a long branch. Okay, so the way to interpret this, and then here even there's in the red population one as well, the same region. The way to interpret this is that a genome region came in by horizontal gene transfer from an unrelated group to those 2 populations and swept in a population-specific manner. So while this population remained diverse, this one has now reduced diversity, basically. Right? And so this, we call these islands of differentiation. The genes have swept populations. They account for the majority of the ecological split signal. And we also detected genes of potential ecological relevance, such as differences in biofilm formation, sigma factors, attachment, and so on. And in subsequent work, actually, we were able to see that the green population doesn't like to attach, whereas the red population very firmly attaches to particles, for example. Now, this leaves us a little bit with this conundrum or this paradox. How can the genome be completely mixed, most of the genome across the populations? And then how can you have at the same time a population-specific sweep? And so Jesse then developed a rationale how to actually record or measure very recent recombination amongst the genomes. And so we had some incidences where we had two nearly clonal genomes that most certainly diverged after the ecological split happened. And he then quantified the number of recombinations that they had within the population and between the population. And then the idea is that if these have enriched within population recombination, then the most recent recombination should already be population specific across the genome. That's indeed the case. That's shown in this graph here. In all the cases that we detected, they had enriched within population recombination. And so the important lesson here is, which is also important in the next section, is that when you use phylogeny, it integrates potentially over a too long time frame to actually really detect population structure. Okay, so that's an important lesson here. And the second one is that you can have these gene-specific sweeps that indicate recent adaptive events in populations, and are also at the same time a really good test for population structure, whether you got the population structure calling right. So then Phil Arevalo, and in collaboration with Jeff Gore at MIT, we asked the question then whether we can actually predict microbial species and populations based on gene flow alone, so in a completely phylogeny-free space. And so the idea is very simple. It just basically says that although everything can in principle recombine with everything, there's structure introduced by genetic similarity as well as habitat similarity. So if you share a habitat, you have more opportunity to share genes with each other. And if you're genetically similar, you have a higher probability of exchanging genes as well. And that probability, in fact, decays exponentially with sequence divergence. So that's a very strong factor. And then once you have detected your gene transfer, you can construct gene flow networks and look for clusters in these networks. Okay, so and then the question was, how do we actually do this? And Phil had a fantastic idea. He said we can divide the genome into, when you look at two closely related genomes, you can divide the genome into segments that are identical and separated by single nucleotide polymorphisms. And then the question is, are these segments that are identical to each other identical because they've been vertically inherited, or whether they are identical because they've been recently transferred amongst the genomes? And how can we actually distinguish amongst the two? And so the idea was actually then fairly simple. So here we have two genomes that are non-recombinogenic. We assumed that all mutations occur randomly across the genome, generated a very simple null model of a Poisson-based null model that just assumes, as I said, random generation of mutations, that then gives you an expectation of a near exponential distribution of identical genome windows. And if you have recombination that introduces identical windows by recent horizontal gene transfer, then you should see a deviation from that expectation. So that's shown here actually. Here's the non-recombinogenic null model. And then here's two genomes that share genome regions by recombination. And then you can measure simply the difference between those curves by some measure. We use just the sum squared, and we call this length bias. And this length bias can then be a edge in a gene transfer network. So two genomes would be nodes, and then the length bias connects these genomes, and is an indication of recently transferred DNA. We validated this with 3 model systems that are ecologically and taxonomically quite diverse. Our own Vibrio system that I already introduced, Rachel Whitaker's Sulfolobos system. These are archaea living in hot springs, where she has defined populations that are physiologically and genetically different, and Pennichism's Prochlorococcus system, where they also spent many years sampling genomes and correlating them to environmental factors, and were able to define populations. This is a busy slide, but I just want to hope I can convince you here that in virtually all of the cases, we were able to recapitulate this population structure that was previously identified. And the surprising thing to me is how well isolated the clusters were that we actually detected from contemporary gene flow. So I think actually that we might be overestimating, by using phylogeny a lot, the actual magnitude of gene transfer that is constantly happening. And so we find these much more isolated gene transfer networks. I should also say that these are consist of strains, at least in our Vibrio system. I lost my cursor, there it is, that we previously identified as parts of populations, and the gray ones are ones that we added new. And the dark lines here the dark and combination of light and dark lines denote all clusters that originate from the raw network, so no clustering algorithm applied. But when you apply clustering algorithms, you find additional structure. And at least in our well-characterized case here, the speciation case, it actually confirmed this speciation event. So we think that these are still connected by some gene flow, perhaps, but you can actually detect these quite well. So then the last question that I want to address now is whether we can then use the same rationale that I introduced previously to see if we can find specific adaptations that indicate selective ecological optimization amongst closely related populations. And then finally, to see basically whether we can improve also not only population identification, but also environmental or health association with these loci that we identified. And so the example I want to bring is also from this paper that Phil and Dave spearheaded, is Ruminococcus nevus that occurs in the human microbiome. There was a previous paper that postulated that there were 2 clades that they found to be somewhat differentially associated with disease. And so we were interested in taking this example and adding some new genomes and trying to do some finer association studies using metagenomes. And so here's these different isolates. You see that they come from different sources. When we apply our clustering algorithm, we basically recovered 3 populations. We do not consider population 3 further because it's consisted only of 2 strains, but it shows you the sensitivity of the method that you can actually exclude at least some strains that are at least doubtful in their association. And now this point that I made before, that phylogeny on this level is actually quite unreliable. When you do whole genome phylogeny and look for support for all 3 populations, you see that the support is actually very low. Although the individual clades themselves have more support, all 3 have actually very low support. And so then Dave developed this based on our previous work, this rationale to actually detect genome regions that have undergone population-specific selective sweeps. And so what he basically said was we need to look for regions in the genome that are monophyletic in at least one of the populations and have reduced diversity. And in most cases, that diversity is near zero. And then once you have this, it provides actually a good opportunity to take all of these regions and use them as quite unambiguous markers to look for the populations in metagenomes. And then the idea is that you can perhaps look for associations better. Okay, so we did this. We called it a reverse ecology framework, where you have closely related isolates or single cell genomes. There's a caveat to that. The Prochlorococcus data set that I showed was single cell genomes, but they were exceptional quality. So for those of you who know Penny Chisholm, know that she will only accept super high quality stuff. And any contamination may result in false clustering, because we've used other single cell genomes and it didn't work quite as well. You generate these gene flow clusters, they are your hypothesis. And then for me, if you can detect actually cluster-specific alleles or genes that have swept, this is not only a very nice way to look at selection, recent selection, but also a very good confirmation that you got the population structure right. Because I can't think for my life of a way that you could see these sweeps that are consistent in different loci across the genome and be an artifact of any kind of false clustering. And then you can map this onto the environment, and that makes them the reverse ecology approach. So we did this with these Ruminococcus guys, and this is now showing these sweep regions. They again, just as a reminder, they have reduced diversity within each population. That is shown here. So these regions, this is the population average nucleotide diversity. And in all cases, these regions have much reduced diversity below the population average. But the really interesting thing is also that in almost all the cases, also it had increased between population diversity, meaning that they also most likely came in by horizontal gene transfer and swept in a population-specific manner, with the exception of this cluster of genes here in population 2 that might have originated from within the population by mutation and then swept through the population. And this is just a more sort of population FST measure, classical population genetics measure. This is just showing you an example of this elevated between population diversity, a couple of regions and the associated trees down here. In the red population, basically, this region was completely unaffected, but in the blue population that you had this increased diversity, and vice versa. Here's an example of the red population and nothing in the blue population. The interesting thing is also, and I took that slide out in the interest of time, but I will mention it still, is that these regions are oftentimes very small. They affect single genes and sometimes only domains of genes. So selection seems to be very highly precise here. And we don't, at least in this case, we didn't see that there was large hitchhiking of neighboring regions in these sweeps. Whereas with sort of this everyday horizontal gene transfer and neutral recombination, you can get a lot of hitchhiking, obviously. The interesting thing was that it did increase the resolution in association. So the population 1 was ever so slightly more associated with healthy individuals, but population 2 was quite a bit more associated with Crohn's disease patients, but not ulcerative colitis, interestingly, and was also less abundant in healthy individuals. So basically, just to summarize this part of the talk then, we hope that this might help to resolve association studies a little bit better, particularly in the human microbiome, where this is very important when you have health and disease, oftentimes ambiguous associations. And then in general, this reverse ecology framework, if you're interested in how organisms adapt and what really the interacting units are in environmental samples, this might be a good approach to actually try and figure this out. And again, you cluster the genomes by this new measure of gene transfer, look for sweeps, and then use these sweep regions to figure out potential adaptations. And I should also say that to me, this is a potentially interesting avenue because these regions are very difficult to obtain in forward genetic screens because there are oftentimes only allelic differences, right? And so it would be difficult to find good phenotypes perhaps in these screens. But here you could target them and exchange alleles, for example, to test hypotheses of effects. Okay, so I thank you with this, and I want to thank our funders, NSF and the Simon Foundation recently. And again, if we have time, I'm happy to take more questions.
Thank you so much, Martin. This was a great talk. I do have one question. So do you see that these sequences that differentiate populations often play a regulatory role in some way?
I'm sorry, I didn't catch the last part. Okay.
Do these sequences play a regulatory role?
You know, unfortunately, and again, a case where in the Ruminococcus where we really looked in more detail, they annotated very poorly. A lot of membrane stuff, though, transmembrane stuff. In the Vibrio system, we were a lot more lucky, and it was really so neatly differentiated into biofilm formation versus non-biofilm formation. So this red population had the MSH pilus that cholera also has to attach. They form thick, juicy biofilms. The green population doesn't, is a good swimmer, and is very quick to swim around and swim between resources. And so we worked with Roman Stalker, who did these microfluidics experiments, and he could really show that this is a very nice ecological trade-off, that the one attaches and firmly, you know, exploits these particles, and the other one just swims around and takes up dissolved organic matter. And so this is something that I should mention also. You need a trade-off to introduce a gene flow barrier through these ecological interactions, right? Because otherwise you just get the genes will spread in the entire population. So we think that a trade-off, you get some adaptation that introduces a trade-off, and you're no longer as good in your ancestral habitat, but you're much better in the new habitat, and that can lead to a physical separation of the gene pools that is then enforced by mutation and recombination.
Thank you.
Emily asked a question here. Given what you said about the difficulty in characterizing certain phages with a standard storage and extraction practice, do you have advice for people on how to store and extract DNA or samples to enhance diverse viral detection?
Yeah, that's a great question. I mean that, you know, there's a DNA storage. There's a lot of papers that have been published about that. I mean, we freeze everything when we can. You know, we did try to extract the DNA as quickly as possible and then freeze everything in the -80. But for these viruses that I mentioned before, you need to proteinase the DNA. That's the only thing I can add, perhaps. That proteinasing, it might not be a bad idea, because if you have covalently linked proteins, as we had in this case, then the DNA will partition into the protein fraction. Did that answer your question?
I think so, yeah.
Okay, good.
I had another phage question. Did you see any correlation at all between the rate of replication and the host range of the phage?
That's a really good question. No, not really. We haven't. I mean, with our data, we can't resolve really the rate of replication very well in our data. That's something I'm interested in, but there was another broad host range phage that we pulled out that's extremely large. Has like some, I forget now, maybe some 20 tRNAs. And so that one is interesting because like many T4 phages, it cuts up the host DNA and then provides its own, its own tRNAs to keep the translation going. But that was literally the only other significant broad host range phage. All the other tailed phages were just super specific.
Okay.
And did you use specific mechanism for isolation of this broad host range phage, or they were like broad host range phages by nature?
Yeah, I mean, you can think of a number of different things. I mean, the one thing I would suggest is make sure your plates are in good shape after 2 weeks and keep watching them. The plaques are small, so you have to use probably a microscope, a stereo microscope, and you could also potentially use perhaps density centrifugation. They might co-purify with vesicles. They look a lot like vesicles, I may add, if you're not very careful in the EMs that you do. So there's another confounding effect. And so if you see DNA in vesicles, it doesn't necessarily mean it's DNA from the host. It could also be these types of phages, because there's a lot of prophage, and they excise at a considerable rate in just regularly grown cultures.
That's great. Well, the last question that I have: how frequently you isolate jumbo phages, like the phages that have a genome size of more than 200 kb?
Yeah, it's rare. It's rare. So. At least amongst bacteriophages, these are very rare. You have to go to eukaryotic viruses to find those really large ones. We just started working on those, and they're super interesting.
Yeah.
Amongst bacteria, I would say that most of the tailed phages are, on the order of 50 kb or so.
Thank you. Very interesting.
Yeah.
So one final question, sorry, for coming from Kandil. So he said, you showed agro-EL phylogeny. Is it involved in any way in phage resistant or susceptibility?
Yes, actually, that's it's so interesting because there is data on that. We haven't specifically looked at that, but this guy, I forget his name. He did his PhD with Luria at MIT many years ago, and he just wrote that during his PhD he isolated phage and host, and the phage had heat shock genes in them, and they could actually overcome resistance. So I think there's some interesting connection, but we've never really looked at the details. But we're really working on making this phage collection accessible broadly, because it's fully genome sequenced. We have the hosts. We've sent it to many different people already, and I think there's if you just clean the genomes, that you find all sorts of really interesting connections, and so we're happy to send them if anybody's interested. Once we can go into the lab again. So.
I have a quick question about the population methods that you were talking about. We have a multipartite genome. I work on Inferred and Rhizobium, or well, whatever you want to call it. And I was wondering and we have 3 subgenomes that all sort very independently of each other. And I'm wondering if that if using your method with that sort of subgenome structure.
When you say sparse, what do you mean?
Meaning we have a megaplasmid and then we have a main chromosome, 2 megaplasmids, and when you look at structure, they have completely different linkage between these subgenomes.
Yeah, that can be a problem. I would honestly, so our method is really, designed to look at very closely related things. I should have probably mentioned that, be more clear about this. If there's already structure in the 16S ribosomal RNA, our method results in trivial clustering, for example. So we target things that are nearly identical in the ribosomal proteins. So that said, I think the method might still work. There may be different history between the plasmids, obviously, and the genome. If you can resolve it well, I know that many plasmids assemble very poorly, but if you can resolve it well, I would try with and without plasmid, for example.
Yeah, well, and the plasmids are almost as big as the main genome itself.
Yeah.
Okay, great, thanks.
Yeah, no, I know that rhizobia have these oftentimes crazy many plasmids, and I've worked a tiny little bit on some of them, and they have very different history, and they travel, for example, amongst different strains and so on. So these mobile genetic elements have a whole life of their own, and if they're very big, obviously they can introduce a connection that is not really in the genome, right? So what we do also now, I should say, that's not in the original paper, but what we do now, we make sure that the genome alignment amongst the strains that we look at is of a considerable size, because otherwise, you get these spurious connections by mobile genetic elements sometimes.
Yeah, it's a huge puzzle to kind of figure out because there's a completely different signal between all of them, and yet they all have identical 16S. They're very closely related to each other, and yet the population histories of each of those.
Yeah, I'd be super interested in if you want to write to me, and because I have a postdoc who's really streamlined the analysis pipeline a lot.
Oh, that'd be great. Yeah, I mean, we have over 600 strains fully sequenced now.
Cool. Yeah, that sounds great.
Okay, great. I'll contact you.
Through all the human microbiome genomes. What's another 600 strains?
Awesome. Thanks.
Thank you so much for this talk.